Hello students, in our yesterday's class we discussed about the protein, then what mean by protein, what are amino acids, which bond is present in between two amino acids, what mean by heteropolymer, so and what are essential amino acids and what mean by non-essential amino acids, and also we discussed a very important topic, that is functions of proteins, that is it play a very important role in the transportation of nutrients across the cell membrane, then enzymes, then hormones, then collagen, rubisco, etc. So in the same time we also discussed about one more topic, that is polysaccharide, long chain of monosaccharide units. It may be glucose, it may be fructose, it may be galactose, it may be ribulose. Many different monosaccharide units are there. Example, starch, glycogen, then one more is inulin, then many different examples are there, no doubt. And also chitin, N-acetyl galactosamine, glucosamine. Yes, many different monosaccharides units are there. In that, some are homopolysaccharide, some are heteropolysaccharide. I told you the difference. And when you take a long chain of monosaccharide units, in that the right end is called as reducing end and the left end is called as non-reducing end. And also one more important topic is that sometime when you consider the long chain of the monosaccharide units, sometime it may contain the branch or the side chain. Yes or no? So example, starch. In that somewhere it will be having the iodine or I2. So consider, which is blue in color. So this is about polysaccharide. And today we are moving on to the next one more topic, that is nucleic acids. Already I told you people. And those nucleic acids are DNA and RNA. Yes or no? So here the word DNA refers to deoxy. Simple word. Deoxyribonucleic acid, and this is ribonucleic acid. So that deoxy refers to removal of oxygen group at second carbon atom. Yes, at second carbon atom. So it is considered as deoxyribonucleic acid and ribonucleic acid. So when you take this DNA and RNA as the nucleic acids, these are made up of what is the different sentences. Yes sir? So these are nothing but long chain of, that is long chain of nucleotides. Long chain of nucleotides. Long chain of nucleotides is also called as polynucleotides. It is also called as polynucleotides. It refers to many nucleotides. So many nucleotide units is considered as polynucleotide or nucleic acid, that is DNA and RNA. So in this, what are the different things or the chemicals present in the nucleic acids? I told you very in detail. So there are three things. One is that is sugar. That sugar is considered as the sugar which is present in the nucleic acids that contain 5 carbon atoms, so that the carbon which contain 5 carbon atoms is considered as pentose sugar. So that pentose sugar is also called as ribose sugar. Yes, it is also considered as ribose sugar. Ribose sugar nothing but pentose sugar, and which is nothing but like this. Yes, this is considered as pentose sugar, and 1, 2, 3, 4 here CH2OH, that is 5th carbon atom. So considered as sugar. And second most important ingredient is heterocyclic compounds, also called as nitrogenous bases. Heterocyclic compounds also called as nitrogenous compounds. These nitrogenous compounds are of two types. One is, I told you, purines, another one is pyrimidines. So purines are of two types. One is adenine, represented as A. Second one is guanine, represented as G. Yes, this is what a recap, because already we discussed in our earlier classes. Pyrimidines, one is cytosine and thymine, that is T, and also one more is there, that is uracil, that is U. So these are what? Four different nitrogenous bases or heterocyclic compounds. In this, these two are considered as double ringed structures, I told you. Yes or no? And these two or three are nothing but these are single benzene ring structures. So this is what the thing. And the third important ingredient is phosphoric acid, or simply we can call it as phosphate. Yes or no? So in this, how can we write the nucleic acid? See, very simple to write. Yes, this is what? The sugar, that is pentose sugar, or we can call it as, yes, right, you can write pentose sugar or ribose sugar. So here at first carbon atom you will be having nitrogenous base. Nitrogenous base, I can write like pentose sugar plus any one of the nitrogenous base, which yield what? Imagine nucleoside, which give nucleoside. Yes, so example, pentose sugar, as it is pentose sugar, nitrogenous base, instead I can put adenine, which yield adenosine. Yes or no? So adenosine, again pentose sugar plus guanine, guanosine. Is pentose sugar plus cytosine, which gives cytidine. Pentose sugar plus T, that is thymidine. Very simple to understand. And these four are nothing but nucleosides. And same time, when we have pentose sugar plus nitrogenous base, at third carbon atom you will be having phosphate or phosphoric acid in the form of phosphate. Now the structure is considered, that is nucleoside plus PO4, which gives nucleotide, refers to pentose sugar plus nitrogenous base, which gives nucleoside. Nucleoside plus HPO4, which gives nucleotide. Likewise here, same time, adenosine plus HPO4, phosphoric acid, which gives which nucleotide? Adenylic acid. Here HPO4, guanylic acid plus HPO4, cytidylic acid plus thymidilic acid. So these are nothing but, see once again, pentose sugar plus nitrogenous plus nucleoside, nucleoside plus HPO4, nucleotide, in that adenylic acid. So the adenylic acid present in the DNA are called as deoxyadenylic acid, deoxyguanylic acid, deoxycytilic acid, deoxythymidilic acid. In case of RNA, so nothing but they are ribonucleic acid, not deoxy, ribonucleic acid. So they are nothing but adenylic acid, like this. So these are nothing but nucleotides. So when we have nucleotides, I mean these nucleotides are arranged in chain, so that is nothing but nucleotide, that is nucleic acid. So I can show you by simple diagrammatic representation, very simple representation. See, we have pentose sugar and here we have adenine, nothing but example adenine, so simply called as nucleoside. Yes, nucleoside. And at third carbon atom we will be having PO4. So now the structure is considered as, the whole structure is nothing but nucleotide. This is one nucleotide. Again you will be having, yes, again one more pentose sugar plus you will be having guanine. Yes, here you will be having HPO4, nothing but one more nucleotide. Again yes, pentose sugar plus HPO4, yes or no, nucleotide, like this. So one nucleotide, here we having a bond, second nucleotide, again bond, third nucleotide, like this. So nothing but polynucleotide. That polynucleotide refers to, that is nucleic acid, which gives nucleic acid. So any doubts in this? Please do remember the concept. So the DNA or RNA is made up of the long chain of nucleic acid. Only one thing, I mean the difference is that in case of DNA the thymine is present, in case of RNA instead of thymine the uracil is present. Uracil is present in case of RNA, instead of uracil the thymine is present in the DNA. This is fundamental difference, so called as nucleic acid, DNA and RNA. So these nucleic acids are mainly present within the what cell, that too in the nucleus. They play a very important role in the they act as a genetic material. They act as a genetic material. They can transform the information from one generation to the next generation. This is what the nucleic acid concept. And after the nucleic acid, again you will be having one more topic, that is structure of protein. So we discussed about the protein, no doubt, but here we are moving on to the next topic, that is structure of protein. Is very simple to understand. Next topic, structure of protein. So already we discussed about what is protein. So that is a long chain of amino acids. Yes or no? Long chain of amino acids is called as, so that is protein, also called as polypeptide. Yes or no? Polypeptide. Many units of amino acid is called as polypeptide. See what happens, means how we can categorize or classify the structure of protein. So based on our convenient, so we are going to classify 4 structures. One is primary structure of protein. So simply I can write primary. Second one, secondary structure of protein, that is like this I can write. And tertiary structure of protein, and last one is quaternary structure of protein. Yes, how, based on what criteria we are going to classify these 4 structures? So simply when you take a long chain, in that imagine we have a protein, that protein is made up of a long chain of amino acids. In that, so one amino acid we have a peptide bond, one more amino acid, peptide bond, one more amino acid, peptide bond, one more amino acid, peptide bond, another amino acid, and last amino acid. Imagine. So when you take this one, in this, imagine it is left side. Imagine it is left side. I mean to say that the first amino acid is always present in the left side. Clear? The first amino acid, imagine it is in this a long chain, the first amino acid is at left side, so that this is represented by N terminal. Do remember this point. So it is considered as N terminal, and the amino acid which is present in the right side is, when you take it as a line, when you take it as a line, in that line, the line starts with the first amino acid, that is left side, and the last amino acid, that is right side, is nothing but last amino acid. So the last amino acid is represented by C terminal. C terminal. How, sir? Based on what? I can give the simple structural formula. See, here we have a carbon, here we will be having COH, H aryl group, and yes or no? NS2 or NS3, whatever you write, NH2. Yes or no? This is one typical amino acid. Here we will be having a peptide bond, one more carbon, again hydrogen, aryl, and COH. Here we will be having NH2 group. Yes or no? Called as a dipeptide. We have a long chain. Imagine long chain. In that, this is, I mean, in long chain, this is nothing but what? Left side. Nothing but first amino acid. First amino acid. In that, it is represented as N terminal. Why? Because we will be having N group, nitrogen group, so called as N terminal, and this is nothing but last amino acid. This is nothing but last amino acid, and last amino acid is present at right side. It is at right side, so that is represented by C terminal. Why? Because you will be having COOH, carboxyl group, that is COOH. Clear my point? So when we take a line of amino acid, long chain of amino acid, left side amino acid called as first amino acid, represented as N terminal, and last amino acid is at right side, represented as C terminal. This can show how. Very simple to understand? Yes, definitely. And in this, see, a small topic, that is, come to this point. What is primary protein? What is primary protein? See, primary protein nothing but when the amino acid, so first amino acid, imagine peptide bond, second amino acid, and third amino acid, and fourth amino acid, fifth amino acid, likewise you will be having 100th amino acid. See, here in a long chain of amino acids, I mean in a protein, the many amino acid units are arranged in a chain with the help of peptide bond. This is called as peptide bond. This is called as peptide bond, so called as primary structure of protein. What is primary structure of protein? In a protein, many amino acid units are arranged in a chain with the help of peptide bond, called as primary structure of amino acid or primary structure of protein. Clear first point? So in this, so there is no alternate chain, there is no branch, nothing. Simply many amino acids are arranged in a chain with the help of peptide bond, called as primary structure of proteins. Very simple. And biologically, these primary structure of proteins are inactive. They do not perform any function. Please do remember, these primary structure of amino acids never perform any function. Yes. Next, second one, that is secondary structure of protein. What makes the secondary structure of protein? Very simple. So when we have a long chain, when we have long chain. I mean to say that we have a long chain of amino acid, in that it is N terminal and it is C terminal. Again we have a N terminal and we have C terminal. Yes, in this we have first amino acid, second amino acid, third amino acid, fourth amino acid and fifth amino acid, whatever the number. Imagine. So this is called as primary structure of protein. Yes or no? Primary structure of protein. So in the primary structure of protein, again we have one more primary structure of protein, that is N terminal and C terminal. So again first amino acid, second amino acid, third amino acid, fourth, fifth, so on. Clear my point? In this concept we have two long chain of amino acids, nothing but we have two primary structure of proteins. So in this, these two are held together by, getting my point? See here only one chain is present, but in case of secondary structure these two chains are held together by, again we have a bonds, we can call it as hydrogen bonds. Clear my point? So now it can make secondary structure of proteins. I hope you understand. Two primary structures together, secondary structure. So these two long chains are held together by hydrogen bonds. So this makes secondary structure of protein. Very clear to understand? Next we have tertiary structure of protein. Next we have tertiary structure of protein. So what it makes? Very simple. Again this is what? N terminal and this is C terminal. We have amino acid, again amino acid, amino acid, amino acid, amino acid, amino acid. And the same time, so one more amino acid chain can overlap the first amino acid chain. Yes, again here we have C terminal or N terminal, whatever. See, again one more C chain, again amino acid, amino acid, amino acid, like this. Clear? So this makes tertiary structure of protein. So better I can tell you people, here one amino acid can wind like this. Yes, N terminal, C terminal. Yes or no? Here somewhere there is a relation between, imagine it is fifth amino acid. Fifth amino acid, same chain, here we will be having 40th amino acid. There is a relation that makes a bond. Getting this? That makes a bond. Again come to the point, here we will be having 30th amino acid, 30th, and this makes it which contain 57th amino acid. Imagine, say 57th. Again there is a relation that makes a bond. Again it will come like this. Yes, getting this? So this is what? The tertiary structure. Here you will be having one more, that is like this, that is N terminal, C terminal. That makes better word I can tell you people, the amino acid chains can wind, the amino acid chains can overlap like woolen bulb, clear, or woolen ball. So consider as tertiary structure of protein. Very simple to understand. And last one is that, here I can write last one, that is quaternary structure of protein. In the quaternary structure of protein, two or more, do remember, use the word two or more what? Polypeptide chains are involved. Yes, in quaternary structure of protein, two or more polypeptide units or protein units are involved to make a fragment, to make the complex protein, that makes quaternary structure. So the best example is human adult hemoglobin. Human adult hemoglobin. So the human adult hemoglobin is made up of 4 polypeptide chain. Made up of 4 polypeptide chain. So those 4 are 2 are alpha chains and 2 are beta chains. Yes. In case of adult hemoglobin, 2 alpha chains and 2 beta chains. That makes here 1, 2nd, 3rd and 4th. NC, NC, again NC, NC, like this. So in this 2 alpha and 2 beta. So that makes quaternary structure of proteins. So here we discussed about structure of proteins. Protein means long chain of amino acid, that is polypeptide. In that, primary structure of protein, second structure and quaternary. Primary means long chain of amino acid, over long chain of amino acid. In that, 2 amino acids are held together by peptide bond. Secondary means 2 polypeptide chains, like this. 2 polypeptide chains or 2 chains are held together by hydrogen bonds. No doubt here will be having a peptide bond, but along with the peptide bond, 2 chains are held together by hydrogen or disulfide bonds. We can call it as disulfide bonds. That makes secondary structure of protein. And third one is, see, 2 polypeptide chains or 2 amino acid chains can overlap each other, and they are most biologically active. Biologically active. So I told you people, primary structure of protein never perform any function. Those must undergo some structural modification. So that structural modification means that tertiary structure of protein. Tertiary structure of protein. So tertiary structure of protein is highly biologically active molecule. Tertiary structure of protein. In tertiary structure of protein molecule, 2 amino acid chains are the overlap each other like woolen bulb or woolen ball. So this is what tertiary. And last one is, in quaternary structure of protein, 2 or more polypeptide chains are involved and they are overlap each other like helically they are arranged. So the best example is adult hemoglobin. Adult hemoglobin. So that made up of 4 polypeptide chains. 2 are alpha chains and 2 are beta chains. This is what quaternary structure of protein. So this is enough for today's class, and remaining things we will discuss in next class. Thank you very much.